So in this video, I'm going to go over a pretty quick pathway called gluconeogenesis. Now, gluconeogenesis is going to be the opposite of glycolysis, where glycolysis allowed us to go from glucose into pyruvate and allowed us to convert pyruvate into a bunch of other compounds like acetyl-CoA and lactate and alanine and a bunch of others. Gluconeogenesis is going to allow us to go from pyruvate all the way back into glucose when we need glucose present in our body. And we're going to see how the same regulatory mechanisms that we saw in glycolysis are also going to be involved in regulating gluconeogenesis, because they're kind of antagonistic, opposite pathways. So when we are performing glycolysis, we're making glucose into pyruvate. We don't want to convert all that pyruvate back into glucose using this pathway. So whatever mechanisms promoted this pathway, they're also going to be suppressing gluconeogenesis. There we go. So these are all the steps associated with glycolysis. And the reason I'm writing this out is actually because gluconeogenesis is going to follow the exact same steps outlined here, except we're going to replace these irreversible steps. There's one there and there's one there. And we're going to replace those with reversible steps that allow us, well, not reversible, but they're going to allow us to go backwards and form these compounds there that we couldn't otherwise get to with the enzymes that we use in glycolysis. And since it's gluconeogenesis, since we're going from pyruvate back into glucose, we're going to start here. We're not going to start at glucose. We're going to start at pyruvate and trace our steps all the way back to glucose. So let's go. To go from pyruvate back into PEP, we're going to need a two-step process, because pyruvate kinase, there isn't really an opposite enzyme that allows us to go the other way. As far as I know, there is no pyruvate phosphatase, so we're going to use a different mechanism to go back. So the first step of what we're going to do is go from pyruvate to a different compound called oxaloacetate. And that's going to be accomplished by an enzyme called pyruvate carboxylase. And it's going to use up an ATP and a carbon dioxide to give back an ADP and a phosphate. So once we get to oxaloacetate, we're going to go to PEP using a different enzyme called PEPCK. PEPCK is similar to GAPDH in the sense that no one ever refers to it by its full name. It's always just called PEPCK, but it stands for PEP carboxykinase or phosphoenol pyruvate carboxykinase. But you can just call it PEPCK, and it's fine. And that reaction is going to take a GTP, which is guanine triphosphate, and we're going to get back GDP and a carbon dioxide. And this is a pretty important regulatory step in gluconeogenesis as well, because pyruvate is going to be present in the mitochondria. So during this reaction, once we get to PEP, this PEP needs to be shuttled outside the mitochondria. And there's going to be a concentration gradient there that's going to control how much PEP we can actually take out. And also, this reaction is not going to be possible, sorry, not this reaction, but it's going to differ depending on the species, because not all species have PEPCK enzymes present in their mitochondria. So humans, we actually have a mitochondrial and a cytosolic form of PEPCK. But for other organisms, this oxaloacetate has to be shuttled outside as oxaloacetate, not as PEP. And we're going to see something else called the malate aspartate shuttle come into play when it comes to taking all this oxaloacetate out of the mitochondria. And that's going to be involved in a different video, because it also has to do with some other metabolic processes, so stay tuned for that. Anyways, after we get to PEP, we can trace all these reversible pathways back. So we can go from PEP to 2PG using enolase, and I know the glycolysis reaction did release an H2O, but I forgot that. So we're going to go from PEP to 2PG to phosphoglycerate, and then we're going to use our phosphoglycerate mutase to move that second phosphate group back into the other oxygen, remember from the glycolysis video. So we're going to use that to go to 3PG. And here, we're going to use phosphoglycerate kinase. Instead of putting in ADP and getting back ATP, that was glycolysis. In gluconeogenesis, we're going to put in an ATP and get back an ADP, because we're adding a second phosphate group. So we're going from 3-phosphoglycerate to 1,3-bisphosphoglycerate. We're adding a second phosphate group there. And then we're going to use our GAPDH. We're going to put in an NADH and get back an NAD plus and a PI. And we're going to get back our, glyceraldehyde 3-phosphate. And remember that in our glycolysis reaction, our F16BP was converted into both DHAP and GAP. using our aldolase enzyme, but we use triose phosphate isomerase to convert all this DHAP into GAP and allow it to go through glycolysis, right? But now in gluconeogenesis, we want DHAP as well. Because of the stoichiometry, we need both DHAP and GAP present. So triose phosphate isomerase is going to help us stabilize our concentrations of DHAP and GAP such that we can go back into F16BP there. But anyways, we're still going to use that same enzyme, our aldolase, to convert both of these compounds back into F16BP. And actually, this is another important point in gluconeogenesis, is glycerol, which is, as you know, a component of our cell membranes, our plasma membranes, is going to enter our gluconeogenesis reaction as DHAP. Glycerol is going to be released when we metabolize our triacylglycerols, or fatty acids, and when that happens, we're going to go from glycerol to glycerol phosphate. I think it's glycerol 3 phosphate as well, using an enzyme called glycerol kinase. And we're going to go from glycerol 3 phosphate to DHAP using an enzyme called, I think it's glycerol 3 phosphate dehydrogenase. Let me write that out. And that's going to get us DHAP. And this is actually something called an FAD-dependent enzyme. It's dependent on concentrations of FAD, which is an electron carrier, but that isn't really relevant for this video. So just keep that in mind, that glycerol does enter this pathway through DHAP. And again, glycerol comes from the breakdown of fatty acids. Because remember, our fatty acids are made up of, well, I keep saying fatty acids, but I specifically mean triacylglycerols. Our fatty acids are made up of a glycerol, and then we have 3 fatty acid chains. And this part there, let me highlight it. Yeah, this part is our glycerol. That's what's going to get us that compound there. Or actually, let me color that in the same color. Yeah. That part of our fatty acids, our triacylglycerols, is going to be our glycerol there. And that's going to go through glycerol kinase to form glycerol 3 phosphate. And then it's going to get converted into DHAP. And that's also going to help us stabilize the GAP, DHAP concentrations to go back in this reaction. But anyways, going back to gluconeogenesis, we're going to come to our second roadblock there, because PFK1 is it's not an irreversible reaction, right? PFK1 can only convert F6B into F16BP. So we need a different compound, a different enzyme to convert us back into F6B there. And that's an enzyme called fructose bisphosphatase. I'm going to write it like this. The enzyme is called, again, fructose bisphosphatase. And that's going to take in a water there, because remember, this is the hydrolysis reaction. We're breaking apart one of the phosphate groups in our F16BP to get F6B. So we're going to have to put in water because it's a hydrolysis reaction, and we're going to get back a phosphate group because we're just yanking it off our F16BP there. And we can always use our phosphoglucose isomerase to go from F6B to G6B, because that is a reversible reaction. And then we're going to come to our final roadblock there, because hexokinase is a single one-way enzyme, I guess you could say. It only goes from glucose to G6B. So we're going to use a different enzyme that we call glucose 6-phosphatase. And I'm going to write that down again. Glucose 6-phosphatase. And that's also going to be a hydrolysis reaction. So we're going to take in water to break that bond, and we're going to get back that phosphate group, and we're going to go back to glucose. And those are all the steps associated with gluconeogenesis. And now we're going to talk about control of gluconeogenesis. So these are all the steps involved in regulation of gluconeogenesis. Remember that we're only going to talk about the 2 major steps involved in regulation. We're not going to go over the whole thing again, but yeah. The first step involved in regulation is going to be the compartmentalization there of pyruvate existing within the mitochondria. So since pyruvate is existing within the mitochondria, we have to convert pyruvate into oxaloacetate within the mitochondria and convert oxaloacetate to PEP in the same place. So pyruvate carboxykinase, pyruvate carboxylase and PEP-CK are both going to exist within the mitochondria, at least in humans, and PEP is going to get shuttled outside the mitochondria to get converted back into 2PG. So all those other enzymes are enolase, our phosphoglycerate mutase, our phosphoglycerate kinase, our GAPDH, our aldolase, all that stuff, they're all going to exist in the cytosol. So we have to transport PEP outside of the mitochondria, and that's going to be a major point of control because, again, the mitochondria, just like any other membrane-bound organelle, has a membrane, and it's going to control the flow of metabolites inside and outside of the membrane, and that's going to be a major point of control for us. And also remember that some species do not have mitochondrial forms of PEP-CK. So this PEP-CK, in humans, we do have both a mitochondrial and a cytosolic form of it, but in other organisms, they only have a cytosolic form. So they have to use something called the malate aspartate shuttle, and we're going to talk about this in detail in another video. But just know that for other organisms other than humans, this oxaloacetate is going to be the one that gets shuttled out, and that's going to get converted into PEP in the cytosol, and then it's going to go through the other the other steps as usual. But just remember that compartmentalization is a huge player when it comes to just any metabolic pathway, but specifically gluconeogenesis. So yeah. The second major step for regulation is going to be our F16BP step there. Remember that this is the same major regulatory part of glycolysis. So this step there, when we go from F16BP to F6B, remember that it was controlled by levels of an allosteric regulator called F26BP, right? F26BP in high concentrations activated glycolysis, right? It activated PFK1 activity, and that means it promoted glycolysis. Right? Remember that F26BP was produced by PFK2, and F26BP activated PFK1, and that promoted glycolysis. Well, when we're doing glycolysis, we don't want to be doing gluconeogenesis, right? If we're going through and we're expending energy to make our pyruvate, we don't want all that to be converted back into glucose. So whatever steps we took to promote glycolysis, those steps are also going to function to inhibit gluconeogenesis there, and that includes F26BP. Now, F26BP is going to inhibit that enzyme that we had there. Let me write that out. We had fructose, I think it's 1,6-bisphosphatase. Yeah. Anyways, F26BP is going to inhibit our fructose 1,6-bisphosphatase because F26BP is going to activate glycolysis through PFK1, and it's going to inhibit gluconeogenesis through fructose 1,6-bisphosphatase. And remember that this was also a major regulatory step in glycolysis. I think I've just said this before, but it's also going to be involved in regulating this. And the other way that we're going to regulate this pathway is through AMP levels. Remember that in glycolysis, AMP was able to bind to the regulatory site of PFK1, and AMP would promote glycolysis because remember, AMP is adenosine monophosphate. Adenosine monophosphate, high levels of it means that we're in a low energy state and we need to make more and more energy, more and more pyruvate. So low levels of AMP would mean we'd have a higher AMP binding to PFK1, and that was going to promote glycolysis, right? But in those same ends, in that same vein, it's also going to promote gluconeogenesis because in a low energy state, why do we want to be converting and expending energy to convert this pyruvate all the way back into glucose when we're in a low energy state, right? We don't need more glucose, we need more pyruvate. So whatever mechanisms we use, whatever metabolites we used to promote glycolysis, they're also going to be inhibiting gluconeogenesis. And yeah, those are all the steps involved.